Okay, hi everyone. My name is Jessica, and I'm the outreach lead of Students versus Pandemics at UCSD. And today we have our speaker, and I wanted to introduce him. He is Dr. Justin Meyer, and he is an associate professor for ecology, behavior, and evolution here at UC San Diego. And I will let him introduce himself further about his research and the topic of this event.
Well, thank you, Jessica, and thank you guys for inviting me tonight. It sounds like a really important group, obviously, during this pandemic and in the future, because we're going to be dealing with issues of pandemics and other diseases and so forth long into the future. So this is a small group, and sometimes when you give these lectures, it can feel like you're just kind of talking into the void of Zoom. Since you're not actually seeing faces in front of you. And so feel free to stop me at any time and ask questions, ask for clarifications, things like that. So I guess to further introduce myself, I study viruses and I study how viruses evolve. And so this is an interdisciplinary field that puts together microbiology, virology, with ecology and evolutionary biology. And where I did my PhD was Harvard Medical School Systems Biology program. So in that program, I also learned these sort of technologies that are used in systems biology and synthetic biology. My research really mixes a lot of these different things together. My goal for the talk today is to give you a little insight into my personal research that I think is most relevant to understanding how SARS-CoV-2 jumped from bats and pangolins into the human population. The actual presentation won't go that much in detail on SARS-CoV-2 itself, but general mechanisms of evolution and things that we've studied in our lab that I think apply to understanding, or at least apply to understanding SARS-CoV-2 and a way to generate hypotheses for how that virus made this crazy jump and has really crippled the global population. I try to pitch this talk at a level that people with not so much biology background could understand, although you might disagree with me at the end of the talk or during the talk. Sometimes we get wrapped up into our fields and we use jargon and so forth. And so I hope to make this as understandable as possible, but please do interrupt me if I'm overusing jargon or saying things that are confusing. And at the end, I hope that we can have a longer discussion about just the details of SARS-CoV-2 and any questions you guys have about that virus or the vaccine that's coming out, what we need to be worried about with respect to the virus's evolution and a vaccine, and so forth. Okay, so I'm gonna get into the lecture now. Okay, so the overall theme of tonight is to talk about how viruses make this transition to infect new types of cells, new hosts, and what are the genetic changes that the virus has to undergo in order to make that transition. And so to sort of talk about that, we have to think about, well, what does the virus have to actually change in order to jump into a new host species? And there's lots of changes that the virus has to make. But one of the major changes is for it to be able to recognize new receptors on the outer membrane of its host. Okay, so let's think about that a little bit more. So this is a diagram of SARS-CoV-2. This is a particle of SARS-CoV-2 entering a cell and then triggering a successful infection. And so tonight's lecture is all on the very first step of the infection, the interaction with the virus with the outer membrane of the host cell. So the first step in any viral infection is what these are called are host recognition proteins. For SARS-CoV-2, or coronaviruses in general, this is the spike protein. And the spike protein interacts with this outer membrane protein. So this is the cell in here. It's trying to get into the cell. And the first step is for it to interact with this outer membrane protein. For SARS-CoV-2, it interacts with the human ACE2 protein. And so it binds to that protein and then triggers the rest of the infection. And so you can think of this protein as kind of a door or a lock that the virus can unlock, open up, and get into the cell. And so this is often referred to as a key and lock interaction. And so therefore, a key step in host expansion is for the virus to recognize new receptor molecules on the outer membrane of the new host. So for SARS-CoV-2, it was to recognize human ACE2 proteins, to recognize this protein on human cells. There's similar proteins like that on bat cells, but they're distinct on humans, and now it has to find that key that unlocks the human version. So in general, how do viruses evolve to use new receptors? How does this process actually work? And so there's a lot of hypotheses for how viruses may do this. And at the heart of answering this question is this more fundamental question in evolutionary biology, that how do organisms in general evolve to do new things, evolve new functions? So when you learn about evolution by natural selection and you learn about Darwin, it's often about how organisms compete and evolve to get better at their current set of functions. It's another question of how do they actually take that leap to do something completely new. And so there's lots of hypotheses that people have come up with. And one that we're going to pursue tonight is one that originates actually in the 1950s. So it's a really old hypothesis. And it's this idea that phenotypic plasticity can drive evolutionary innovation. So these aren't viruses, these are water fleas, these are Daphnia. They're really tiny, they live in lakes all around the world. But they're a really great visual example of what phenotypic plasticity actually is. And so phenotypic plasticity is this idea that a single genotype has the potential to actually make multiple different kinds of phenotypes. That's the plasticity part. Multiple different is the plasticity. And so in this example here, a single genotype of this Daphnia can either produce this form here, which can swim really fast and is very competitive, or it can form this type of Daphnia. So they have the exact same genomes, but very different phenotypes. And this one here is better able at avoiding fish predation. So obviously with those spikes, it's harder for a fish to come up to a Daphnia and eat it. So that's all you really need to know about Daphnia. But the point is that we often think of one genome creating one phenotype, but actually there are ways that a single genome can produce multiple different phenotypes. And so this was known even back in the 1950s, that the genotype to phenotype map isn't hardwired. And so this guy, Waddington, actually came up with this hypothesis that the way that evolutionary innovations worked is that they first experimented with phenotypic plasticity, and then if that experiment was successful and the organism found a new phenotype that worked really well, then it would assimilate that phenotype. It would evolve additional mutations, additional changes that fix that new phenotype. So you could imagine that there was a Daphnia a long time ago, or even right now, that can produce these two different phenotypes. But maybe this one is a lot better because this guy lives in a lake with lots of fish. Then it would accumulate mutations to just always produce this type that's resistant to fish and never produce that type. So this is just giving you this framework of how phenotypic plasticity can work in order to drive the evolution of new functions and innovations. So let's sort of put a pin in that and then get back to my research and get back to viral evolution and how this pertains to host range expansions. Okay, So how does my lab study viral evolution? There's lots of ways to study viral evolution, but the way that we do is through experimental evolution. So what is experimental evolution? It's this idea that you can culture microbes in the lab, and microbes have such short generation times that over the course of weeks and months, you can actually see those microbes adapt and change and evolve during your experiment. And so we can actually culture this virus called lambdaphage in the lab, and we can actually see how it mutates over time, how it adapts to its host. So its host is E. coli, which also is very conducive to lab experiments, and itself has a short generation time and can evolve in the lab too. And so this gives us an opportunity to actually run an experiment and see how these viruses are changing and see how they adapt or see how they innovate to maybe um, use a new receptor or infect a new species of bacteria. And so it's a really powerful system because obviously, we would want to study uh, things like SARS-CoV-2 or coronaviruses, but it's much more difficult because obviously we can't, release a virus into a population of humans or a population of bats or so forth. That would be, obviously unethical. But releasing a virus into a population of E. coli is not unethical. These are controlled experiments, and you can look at the infection of this virus on E. coli. Okay, so the way that the experiments work is that we co-culture these two organisms together, and we let them grow up for 24 hours in just a simple flask environment. And then after 24 hours, we take a portion of this flask, 1% of it, and we just transfer it to a fresh flask. So during that 24 hours, that E. coli is eating resources that we provided in the flask, and then the phage are infecting the E. coli. And so the phage population is growing, the bacterial population is growing. And as this is happening, the E. coli begin to accumulate mutations that confer resistance to the phage, and then the phage accumulate mutations that counter that resistance. And so we often talk about coevolutionary arms races between hosts and parasites or hosts and viruses. Well, we can actually see that take place in this controlled laboratory condition. As we run our experiment, we can preserve each and every day of the experiment. We can create what's called a frozen fossil record. So one of the main challenges in studying evolutionary biology is you often have to rely on modern species to reconstruct the past. However, we can actually reconstruct the past directly because we have this perfect record. We have samples from day 0, from day 1, from day 2, and so forth. And so what's really cool about microbes is that you can preserve them frozen. That maintains their DNA and their RNA, but you can also revive them. These are organisms that can actually be cryopreserved and revived later. And so what I can do is I can compare what viruses and bacteria I put into the beginning of the experiment to the viruses and bacteria that evolved that are now superior and adapted at the end of the experiment. And I can make these head-to-head comparisons of their genomes to find what mutations did they acquire, but also how did their phenotypes change? How did the virus improve at infecting the host cell? So it's a way to actually, perform evolution experiments and see these things happen in action in real time. And this is just to say that nowadays we have lots of great biotechnology. And so if I evolve a virus, I'm able to sequence its genome. So this is just a diagram of lots of sequenced genomes. So the circles are the genomes, and the points here are where different mutations occurred in the virus genome. So these are viruses evolved under one condition. These are viruses evolved under another condition. This is a diagram showing you where the genes in the viral genome actually are. So we don't have to look at very specific details in here, but know that it's very easy to sequence viral genomes now. And so you can find what are the mutations that actually evolved in the virus. Nowadays, we're able to engineer viral genomes really efficiently. And so this is just an engineering strategy that we use called MAGE, Multiplex Automated Genome Engineering. Don't worry about the details. It's just a very efficient way to engineer genomes. And so what we can do is we can say, oh, we think that these mutations here, this seems like a hotspot in both of the conditions for mutations. We think those are mainly responsible for the virus's adaptation. And so now we're going to start over with a virus that didn't evolve in our lab. And we're going to engineer in those mutations, and we're going to see how does that change how good the virus is at infecting the host. And so that's a way for us to test that hypothesis that these are adaptive mutations. And so in my lab, undergrads come in and they decide that they, on a certain set of conditions that they're interested in studying how viruses evolve to those conditions, they run these evolution experiments, they sequence whole genomes of the virus, and they even do the genetic engineering to test out which of the mutations are responsible for the adaptation in the viruses. So it's actually it's really fun to run these experiments with undergrads and see them be able to run through all these different technologies and actually test hypotheses about evolution. Okay, so that's just the general framework of what we do in my lab. Now what I want to talk about is an evolution experiment that I actually did during my PhD that we continue to study because it was such an interesting experiment. And so in the experiment, we evolved Lambda with E. coli. And what we observed is that Lambda evolved from using a single receptor called LAMB. So this is the outer membrane of E. coli, and it has lots of proteins in the outer membrane. One protein is called LAMB. This is a protein structure of LAMB. And it actually evolved to use a completely different receptor, OMP-F. And this is the protein structure of OMP-F. You can see, and I should note that it maintained the ability to use LAMB. Now it's just able to use both of these different receptors. These receptors, when you sort of look at these structures, they look very similar to each other, but actually their underlying amino acids are very different. And so this is a major shift for the virus to be able to do. This is actually a much bigger difference than what SARS-CoV-2 had to do when it jumped into the human population. Okay, so we observed that, how that evolution worked, and so this is a paper from my PhD, is the host, so we're co-culturing the virus and the bacteria together, and the host evolved resistance to the virus. And the way that the host evolved resistance is that it downregulated that LAMB receptor. And so most of the cells in the population no longer had that receptor, and so the phage couldn't infect them. But then the phage begins to evolve additional mutations. These mutations are all in the host recognition protein. That's called J. That's right here. And so they get mutations in the J protein. And then if they get the right combination of 4 mutations, then that virus is suddenly able to use the new receptor called OMP-F. Okay, so that's what we showed. We just basically showed the dynamics in my PhD. And one of the first experiments that we did when I got my lab at UCSD, so this was about 6 years ago, was to figure out, well, how exactly do these mutations work? Yeah, we can identify the mutations, but how do they work at the molecular level? How do they work at the protein level? And importantly, why would they give the virus the ability to use this new receptor? And so now we're going to get into kind of more of the experiments and the research and really trying to understand how these mutations work. And so I'm going to take you on a little bit of a journey here. Basically, we stumbled on this really cool result. And so we were looking at what's called the ancestor virus or the wild type virus. And so this is the normal virus that we start our experiments with. And we were comparing its phenotype to a virus that was able to use OMP-F, so an OMP-F plus virus. Um, and we had noticed that these OMPF plus viruses seemed relatively unstable. What that means is that we would have a culture of them. We'd have no host cells in that culture. And we'd just see that that virus would die relatively quickly over time. So there's no real reason for it to die besides just spontaneously decaying. So, we think of humans, certainly humans have a lifespan. Well, viruses also have a lifespan. This is why, we talk about with SARS-CoV-2, yes, they can transfer from surfaces, but actually, if you let those viruses sit on that surface for a long time, those viruses are going to go away. They're just going to spontaneously deconstruct. And luckily for us, actually, it seems that SARS-CoV-2 is unstable, and so it doesn't actually stay on surfaces much longer than a day or so. Okay, so we were just interested in the fact that this really cool virus that was able to use two receptors seemed very unstable. And so we were running experiments where we're collecting data on just the number of viral particles in a test tube. There's no cells there, they're not growing, they're just sitting there, and seeing how they decayed over time. And so we see that this ancestor has a really uniform rate of decay. Whereas this one that's OMPF plus actually has what looks like at least two different rates of decay. There are particles that are decaying really, really rapidly, and then there's other particles that are decaying really, really slowly. And this was weird because in the literature, most viruses have this kind of pattern of decay. And if you think about it, the viruses, the way that we're running this experiment, they should be genetically identical with each other. So they should all have the same phenotype. They should all have the same death rate. But what it looked like the evolved one was doing is that it actually probably had two different phenotypes. It had one fast decaying phenotype and then one slow decaying phenotype. And given this pattern, what it suggested to us is that there were, even though we had a single genotype, we had some phenotypic plasticity in the virus such that there were fast decayers and slow decayers. And so we thought, oh, that's really interesting. And the reason why we really focused on this, because lots of other people had seen this pattern before, but overlooked it and didn't think that there was much to this pattern. But we didn't overlook it because we were evolutionary biologists studying viruses, and we realized, back in the 1950s, people talked about phenotypic plasticity and how it could lead to evolutionary innovations. And people have brought this up since then. I saw a talk when I was in undergrad, so much like you guys, where somebody talked about this possibility that phenotypic plasticity was key to innovations. And so we were just positioned, it's this serendipity of a prepared mind in order to look at this weird pattern, think that there was something to that pattern, and begin to learn from it. Okay, so this is the key to us figuring out the mechanism of host range expansion. So let's sort of think about phenotypic plasticity and how it might work in terms of proteins. So this is always, when I can think about mechanisms, then I can begin to believe that it might be real, like the things that we're talking about in terms of plasticity might be real. And so the hypothesis is, and I'm sorry, these are graphs from introductory biochemistry class. And so I'm sorry if anybody hasn't gone through introductory biochemistry class. And I'm also sorry for the people that have gone through it, because it might give you some PTSD looking at these diagrams. But basically, in biochemistry, what you learn is that proteins fold into very specific conformations. And what happens in that folding process is that you'll start with a peptide that's unfolded and has lots of free energy. And then that peptide begins to fold in on itself, and it forms a particular structure, and that's a very stable structure. And that structure then gives that protein function. And so our hypothesis is that the ancestor has this J protein at the end of its tail. This is the host recognition protein. And it folds into just one stable structure. And that's the normal dogma of how molecular biology works. But what we think is going on with the OmpF one is that actually the way that it's mutated, it's now in this weird state where sometimes it folds into this relatively stable structure, or it folds into this other relatively stable structure. And so you actually, even though it starts out with a single peptide, it can go down here or it can go down there, and it actually produces phenotypic heterogeneity. And so what we think is that there's an unstable version, a relatively unstable version, and a more stable version, and that's what's giving you the pattern here. And so then the next step of our thinking is, well, this is weird, and it's also weird for viruses to be able to use a new receptor. And so, well, maybe those two things are related to each other. So our hypothesis is that lambda gains the use of a new receptor through evolving a plastic J protein. So the idea here is that this is the ancestral form of the protein. It interacts really well with lambda. That there is this really stable version of the evolved protein still is a stable structure and interacts really well with lambda. But there's a new version of the protein that's relatively unstable, and that this new structure is able to interact with a new receptor that's OmpF. Okay, so that's our overall hypothesis, and it's in line with that sort of historic hypothesis about plasticity. And so, how do we test that for lambda? Well, there's actually a really straightforward experiment that you can run. And the prediction that the experiment tests is that if you start a culture with particles, they should have a mixture of both particles that can use OmpF and particles that can use lambda. And so you'll start out with a lot of high levels of OmpF plus viruses, but because they're more unstable than the lambda viruses, they should drop out of the population so that by the end of the experiment, you'll have very few particles that can use OmpF, but the remaining particles can disproportionately use lambda. And so the null hypothesis is that, well, all particles should be the same. And so even though we have these multiple different decay rates, that doesn't change their affinity for the two receptors. And so you should see the same level of OmpF particles here as you see here. And so what we're going to do is we're going to repeat that experiment where we're just watching the particles die. But we're going to look at how many particles can use OmpF and how many particles can use lambda. In the no plasticity hypothesis, you would say that there should be the same level at time point 0 as time point 1 day. But in the alternative hypothesis, plasticity hypothesis, you should see that ratio will shift so that there's fewer particles on the first day, 24 hours later, that can use OmpF. So that's a really straightforward experiment to do. And indeed, the results are directly in line with our hypothesis about plasticity. So not all of the viral particles are the same in this population. Some of them are unstable, some of them are stable. The unstable ones are also the ones that are able to use the new receptor, where the stable ones are the ones that are able to use the old receptor. So underlying this innovation is this phenotypic plasticity. Okay, well, that's all well and good, but that's just one experiment. So what I want to talk about next are two additional experiments to sort of prove this point that plasticity is important for the virus to use that new receptor. And so the first experiment is very similar to the last one, where we're just trying to establish that there is heterogeneity among particles, some that use lambda and some that use OmpF. Even though they're all genetically identical to each other, they have these different phenotypes. And so we're going to do this experiment that I call an OmpF binding pull down experiment. And so we are just going to take cells and lambda. These are all the ones that are the evolved lambda that can use the new receptor. And we have cells. What's important to note is that they don't have LAMB, they only have OMP-F on the outer membrane of cells. So you can engineer E. coli very easily to knock out genes. And so we have a knockout version of E. coli that only has the one receptor, the OMP-F receptor. That's the new one. And so when we run this experiment, we mix them together and immediately we remove the cells. So it's very easy to remove cells. You just centrifuge them, you put them into the bottom of the tube. The viruses are floating in the liquid in the tube. And so they're really not able to bind these cells because this is just very, very quickly. They don't have the time to stick to them yet. And so they're just free floating. And we take them out and we put them onto bacterial lawns. So these lawns have just different bacteria on them. And we put the viruses on the lawns. And if the virus is able to infect the bacteria, then it produces this clearing that we call a plaque. And so these are all, each of these spots is where a virus found a cell that could infect, create a little mini epidemic and spread in this circular shape. And so what we're going to do is we're going to use cells that only express LAMB or cells that only express OMP-F. And this just gives us a baseline of how many phages are in here and do they have a preference for LAMB or for OMP-F. And so we expect that when we initially start, since they haven't started binding to these cells, there should be a nice mixture between both LAMB cells and OMP-F cells. I'm sorry, LAMB targeting phage and OMP-F targeting phage. Now, what we also do at the same time is we incubate them together for 90 minutes. And so this gives the virus time to actually bind to the cells. And then we centrifuge out the cells, and there's some viruses that are left over that haven't found cells to bind to. And then we plate them onto the two lawns again. And so what we're asking here is, has this been distorted? And so the null hypothesis is that no, you'll see the exact same kinds of patterns as you saw in the initial plates if there's no plasticity. So the idea here is that all viral particles are identical to each other. And so when viruses bind to the OMP-F cells, the remaining viruses will have the exact same affinity for OMP-F and the exact same affinity, for OMP-F or the exact same affinity for LAMB. But the plasticity hypothesis says that no, not all of these viruses are the same. They're not all uniform. And so red viruses, the OMP-F viruses, are going to disproportionately bind to these cells that are expressing their receptor. And they're going to be removed. They're going to be in this pellet at the bottom of the tube. And so when I draw off phages from this tube, I'm going to disproportionately draw off the ones that are focused on infecting through LAMB, and I'm not going to see any plaques on OMP-F, or I'm going to see a reduction in plaques on OMP-F. And so sure enough, when we actually do that exact experiment, this is what the initial plates would look like, and this is what the final plates would look like. Here are the actual data. On the x-axis, we have that 90-minute incubation period. On the y-axis, we have just total number of phages, and that's in the solid line. Those are phages that are able to infect through LAMB, and the dotted line are phages that are able to infect through OMP-F. And so we can actually see that pull-down experiment actually disproportionately pulled out phages that can infect through OMP-F, the new receptor, and that suggests that indeed there's heterogeneity in that population. Not all viruses were the same, and it provides evidence for this plasticity argument. Okay, so we have one more experiment left, really hitting home this hypothesis that plasticity is important for viral evolution. And this is where we get a little bit more sophisticated, where we actually want to engineer the protein. And so remember, this is the pattern, this what we call biphasic decay is the pattern of a virus that has this phenotypic plasticity. And so what we wanted to do, we wanted to find mutations that would take away this plasticity property. So we have a virus that is able to use OMP-F. We want to engineer in mutations to that J protein in order to remove its ability to be plastic. And removing plasticity, we then predict, should remove its ability to use the new receptor. So what we have to do, so we set out to do this, and it was a little bit complicated, but in the end, we used that engineering process that I talked about, and we found two separate mutations that cause the protein to no longer produce that heterogeneity, no longer being plastic. So it has all the mutations necessary to use the new receptor, but one additional mutation that caused it to no longer be plastic. And so we actually found two different ones. And so we made a construct that was OMP-F plus with this additional mutation, or this other construct that was OMP-F plus with this additional mutation. If you're unfamiliar with this nomenclature, it just says that at this site, an amino acid was changed from a T to an A, or at this other site, an amino acid was changed from an F to an L. And so that gives us these, and this is the proof that they're no longer bistable. They have a single uniform rate of decay that's just like that ancestral version of the virus. That's the typical pattern that we see. And so then the question is, we weren't engineering directly, we weren't reversing any of the mutations that gave the virus the property to use the new receptor. We added additional mutations that changed plasticity, and then we took those viruses and we plated them onto lawns of bacteria. And now these are not cartoons, these are real images from the lab, where what we see is that the one that we haven't added any mutations to can produce a really nice clearing on this lawn of bacteria. And the bacteria no longer express LAMB, they're only expressing OMP-F, and so only an OMP-F plus virus is able to produce the clearing. So we see a clearing there, no clearing here, no clearing here. And so it's that really great experiment where you can make a genetic modification, predict that it's going to have this cool effect on its phenotype in kind of two different ways. And it's that final proof that, yeah, even if you have all of the necessary mutations to be OMP-F plus, if the protein doesn't have this plasticity, then you remove the property for it to be able to use that new receptor. Okay, so this is just kind of wrapping up in a cartoon. What we observed is that the virus goes through, this is what you guys have learned is the central dogma for molecular biology, where you have DNA that then expresses RNA, then that RNA is translated into a polypeptide chain, and that polypeptide chain folds into a particular structure. And that structure for the J interacts with this one single receptor. What had happened in our experiment is that this DNA evolved 4 different mutations. It changed the RNA, it changed the polypeptides that form, or the sequence of polypeptides, and then that caused it to have this ability to not just fold into one type, but fold into 2 different types that each have activity on these different receptors. So it's this evidence that phenotypic plasticity is responsible for this new function, this new property. So, I started this out by talking about SARS-CoV-2, and obviously we're all thinking about this. And I have to say that I have no evidence, no direct evidence that SARS-CoV-2 used this mechanism in order to expand its host range. However, there are some early signs in its evolution in humans that actually suggest that this could be possible for this virus. And so the question I want to ask is, was the original SARS-CoV-2 strain phenotypically plastic? And was the original strain unstable? So remember that was the first hint that something cool was going on with our virus. And so what we know is a little bit about an answer to this question here, in that, yes, the original strain of SARS-CoV-2 that jumped into the human population was unstable. And the part of it that was unstable were not all of these other components on the virus, but was the host recognition protein. And so what we see is that people have been studying the evolution of SARS-CoV-2, and there is one particular mutation in that spike protein that has dominated in populations of SARS-CoV-2. So as it's spreading around the world, this mutation seems to be spreading faster than other mutations in the virus. And it's a D to G change at position 614. It's in the spike protein, that's the host recognition protein. And what people have found is that that actually stabilizes the spike protein, and that stabilization is really good for the virus. It helps it specialize on the human ACE2 receptor. So what this tells us is that the virus that spread into humans was not optimally stable. It was actually unstable. And now, as this virus is getting better and better at spreading in humans, one of its adaptations is that it's stabilizing this host recognition protein. What it suggests, this is in line with our model, is that maybe that initial virus was unstable and was phenotypically plastic. And now there's mutations that are doing what we call genetic assimilation, back to that 1950s paper, where they are stabilizing the protein in a new conformation to better use the human receptor. So that's just one little bit of evidence that at least things are in the right direction for our hypothesis to be applicable to SARS-CoV-2 evolution. But of course, we need to do the experiments. And so in my lab, we are teaming up with people. We're doing bioinformatic analyses in order to figure out whether or not that initial strain that jumped into humans had this phenotypic plasticity property. Okay, so that was a lot of talking about my research. And I just want to thank my lab and all of the great people that I've had the honor to work with, and of course funding sources and so forth. Okay, so I'm going to stop sharing my screen right now and answer any questions that you guys have.
I have one question. This could be, I don't know if you've already mentioned this earlier, but I guess like for the plasticity that you were talking about, is it just due to like random mutations, and then since like it gave it like a phenotypic like benefit, it just went like crazy, I guess?
Yeah, no, exactly. So that's a part of the story that I really didn't belabor. I showed that cartoon where I show that, okay, they get 4 mutations in that J protein, and then that produces this plasticity, and that's really good. It allows it to use the new receptor. But why did they get those 4 mutations in the first place? And so that we're still investigating. We know that those mutations are adaptive. We know that those mutations help the virus better find its host cell, but we're not really sure how they work. And the biggest mystery is like, well, why do they create this plasticity? Is there something inherent to like the way that they're being selected on that also favors them to create this plasticity, or is it just kind of some random effect that is hard to that just is kind of lucky for the virus for it to happen? So. Yep. No, really good.
And I guess like a follow-up, I guess a question to that is like, I know in the beginning, like you were like, we will start like have to better prepare for like all the other future viruses that might come through and etc. So if this is like, I don't know if there's a real way we can prepare, but if everything is due to like this whole like random chance, like I guess like how would the preparation aspect occur? Because we like even see this with now, like no one thought it would just be like this gigantic pandemic, because all these mutations are to a random effect. So like if we were to prepare for like something like this happening again in the future, like how would the science aspect of that be?
So there's really great research being done by different groups. There's one that's based out of New York, Global Health Alliance, I think, or I'm forgetting the name of it right now. But what they're doing is they're going over to China and to other places where they have predicted that there might be pandemics originating. The reason why they've chosen these locations are they have a statistical model that incorporates a lot of different factors. But one of the factors is encounter rate between humans and potential viruses from things like bats or pangolins or chimpanzees are the source of HIV. And so their idea is that they want to sort of understand these viruses, understand what characteristics these viruses have, and what are the characteristics that allow them to jump into human populations. So I would pose to them that, oh, I think it's this plasticity, and so are there certain genotypes of viruses that have that potential? And what they do is they sequence their genomes and they identify them and so forth. And so perhaps they could actually identify viruses that had the potential to jump into humans and intervene before they actually did. So that's sort of one way, but that's really advanced. That's, you know, a decade away for us to be able to really do that. So in the short term, you know, that statistical model has a bunch of factors that make people more at risk to the emergence of a new disease. And so some of those factors are, you know, human encounters with animals, and so that is, you know, they focus in on, say, the exotic pet trade or the bushmeat trade or things like that. And basically their idea is like, just leave animals be where they are, and, you know, just try not to invade their habitats, and then that will reduce our exposure to the diseases that they have. So that's one way. There's lots of other ways. They predict that you know, forest destruction is bad, in that animals, once their habitat is destroyed, have to move into other locations, and that movement, that migration, enhances our ability to, or enhances our potential to interact with them, and then to catch their viruses. Other things are, you know, we are going to, so that's sort of at the emergence end, but also, you know, this virus really took off and spread around the globe. You could imagine having better sort of warning signs that there's a new virus, and for governments to take this a little bit more seriously, especially in the United States, where we haven't taken it so seriously, and, you know, listen to public health officials and clamp it down immediately. Certainly other countries that had experienced SARS-1 were much, you know, better at responding. And, you know, in you know, Hong Kong and South Korea and tons of other countries did a much better job than the United States at dealing with this immediately. And, you know, their economies are up and running, their people are healthy, haven't suffered the same death. So there's things on the front end, but also once it does start spreading, that we could begin to do. I teach this evolution of infectious disease course, and I've taught it for 5 years. And at the beginning of each time I teach it, I give a lecture about, you know, all of the different factors that are all the different ways that the globe is changing that have enhanced the ability for a pandemic to happen. And I warn students that this is going to happen if we don't change these things. And I never thought that I would be teaching through a pandemic. This year, you know, that slide was hard, because it's like we predict every year, and I don't want to be right about this. But it, you know, and others, I'm not the only scientist that predicted this, that something like this would happen. And actually that group that I was talking about, they were studying coronaviruses in Wuhan, and you know, had cornered that as a potential place for a pandemic to start. So there is we could do better than we have done, for sure.
Yeah.
So somebody is raising their hand. So yeah, Celine.
Hi, I just wanted to say thank you so much for the presentation. It was really interesting and I learned a lot from it. I actually had 2 questions. The first one regards more to like coronavirus specifically. I know you were talking about like how COVID-19 infects specifically human ACE2 receptors. Is that correct?
Yep.
I know that when someone is like infected with COVID-19, it's mostly like respiratory symptoms, but I know that there have been cases where people have been getting strokes and just like a lot of symptoms like not related to the lungs. So are ACE2 receptors like present like throughout the whole human body?
They are, and I don't know exactly the distribution of, you know, which cells have them and which cells do not. But we do think of it as a respiratory virus in part because of the way that it spreads. You know, we breathe it in and then it gets into our, but actually it spreads in our blood. And so it's, people are actually thinking of it as more of a pulmonary virus, or a pulmonary infection, where it spreads throughout your circulatory system. And so people have these weird, you know, there's this thing called COVID toe. I don't really know what that is. It's just, but it's this weird symptom that people have with COVID-19. And it turns out, I think it maybe they bulge, or I'm not I wouldn't Google search that. I'm sure it's not going to be like a nice picture. But that it turns out that the reason why that happens is, you know, your toes are furthest away from your heart. And so if you have circulatory problems, that's where they begin to show up. And so the virus is actually damaging not just the lungs, but the circulatory system as well, the pulmonary system. And that's why people also have fogginess, you know, because they're just not getting as much circulation to their brain and so forth. And, you know, there's all kinds of other, and strokes, like you're saying. So it's a pretty nasty virus in what it does to our bodies, definitely.
Yeah, thank you so much. Yeah, that makes a lot more sense. The other question that I had was, I remember at the very beginning of the lecture, you were speaking about just like the coronavirus like vaccine that's coming out with like a lot of news. So I guess like how do like potential treatments and like vaccines that are coming out with like Moderna, how does that account for like mutations with COVID-19?
Yeah, so luckily, okay, so vaccines, the way vaccines work is they present, you know, typically a protein to your immune system. Your immune system learns that protein and learns that it's bad and kills off, you know, but maybe binds up the protein. And so if it's like the spike protein on the coronavirus, then it's going to block its ability to infect cells, and so it's going to be effective at wiping it out. And so, but the problem with that is that the viruses mutate and their proteins change. And so if they change significantly enough, then the vaccine is going to be mismatched to the actual protein in the virus that's spreading. And so this is what happens in seasonal flu, is that there's just, seasonal flu has a really high mutation rate, and there's a lot of genetic diversity in the flu. And so it changes really rapidly. So from one year to the next, you have a completely new strain, different proteins, and your immune system doesn't recognize it. And so your immune system has to relearn to recognize it. And this cycle happens again and again and again. And this is why each and every year we need a brand new vaccine for the flu. It's also why we're bad at predicting what strain is going to be infecting us, because it's changing so fast. And so we make a guess, but sometimes that guess is wrong. And so some years the flu vaccine is not very effective because it's presenting the wrong protein. Other years it is more effective. So lucky for us, so both SARS-CoV-2 and influenza are RNA viruses. RNA viruses do have a higher mutation rate than DNA-based viruses, and then we're DNA-based as well, of course. But it turns out that the SARS-CoV-2 and all coronaviruses, they have an extra protein that helps with proofreading, so that when they replicate the RNA, it can detect whether or not a wrong nucleotide was put into the RNA, and it can fix it. And so this causes the SARS-CoV-2 to have a relatively low mutation rate compared to influenza. And so, yes, this thing is mutating. Yes, this thing is evolving, but not nearly as fast as influenza. And so what that means is that the vaccine that we create today will likely present a protein to our immune systems that long into the future will be the same type of protein or very similar protein as SARS-CoV-2 in the future. And so the vaccine today will likely maintain its efficacy. And we learned, you know, recently that the efficacy is 95% for both the Moderna and the Pfizer. So the Pfizer, there was new data today that said, before they said above 90%, and today they said it was 95%.
Yeah.
So that's really good, and I bet this will be maintained for a long time. Both of these use mRNA technology, and I'm talking about, the vaccine's presenting a protein. mRNA is not a protein, but what the mRNA does is it gets into your cells. Your cells create the viral protein, and then your immune system learns about that viral protein that the virus didn't make, your cells did. So it's pretty cool that way. And these are new advanced vaccines that are really, they're easy to synthesize. So right now we probably only have about 10 million doses of the vaccines made, but hopefully in a couple months we'll have hundreds of millions of them. It's a lot easier than typical vaccines. And it has other properties that hopefully in future pandemics, since we've proven this mRNA technology works, we can be even faster to create a new vaccine using this technology. So, yeah.
Thank you so much.
Yeah, no. What questions?
I had another question to ask. I was wondering, like, if there are mutations to like the virus itself, like how does that contribute to like if there's like a different like severity of symptoms, I guess, like if it gets like more severe?
Yeah. So there's that mutation I talked about that stabilized the virus. It stabilizes the virus. It causes the virus to actually produce more particles. So the idea is that if it's stable, then it can just produce more viable particles. If it's unstable, they fall apart, and so there's fewer of them. And so that actually causes it to be more infectious, to spread. At least they showed this in cell culture. It spreads faster in cell culture. And then if you look at the data from the spread of the virus around the world, the ones with this mutation seem to spread faster from one continent to the next or one region to the next than other viruses that don't have this mutation. And so that's consistent with it being more infectious. Now the question is, so it appears that it's more infectious because it produces more particles. So does that also have a bad effect on the human body? And so that has not been proven yet. So the fear is that there's a mutant virus that would be worse, that would cause us to be even sicker. That hasn't been proven, but I don't know. I mean, if I had an infection and if I had to choose between a strain that produced only 100 new particles versus 1,000 new particles, I would choose the strain that produced because it's just like a numbers game, right? The more particles, the more cells it can spread to more cells, more, could spread in your circulatory system faster. So we haven't made those connections. That would just be a hypothesis. And I don't want to alarm people that there's a new, even deadlier version spreading around the globe. But I think we need to ask those questions. We need to get the data. And I would have to just say that I bet that what's really bad is getting the coronavirus. And what might be like a smudge worse is getting this mutant coronavirus. Right. And so like in the scheme of things, no, it's the virus is what's bad, not the mutant virus. It's probably just maybe a little bit worse. But there's probably a million other factors that play into the severity of the disease.
Thank you so much for answering my question. I think it's really interesting to see that and to have like the hypothesis as well. Thank you.
Yeah, no, definitely.
I have another question. So like right now, for like the longest time, vaccines have always been like this like debate if they are good or not. Like some people don't believe in vaccines or don't get vaccinated. So with these new vaccines, like in a world where, everyone has equal access to this vaccine, like how much of the population is, like, 'cause I know like herd immunity where it's like, if most of them have it, then like the rest of the population is okay. So I don't know the statistics on how many people believe vaccines are helpful, but it's like, how many people actually need to receive this vaccine so that if there is a good number of people who don't, it is okay?
Yeah. So that specific number is hard to predict. The reason why is that, if we continue to socially distance and we continue to wear masks, then that fraction of the population that needs to get vaccinated in order to drive the disease out of the population will be a lot lower. Fewer people will have to get the vaccine. But, we've done a really bad job in the United States. And so, I think you can't count on that. And so you're going to need at least half the population to get this vaccine. But you're right. There's very good math. We teach it in my course, where you can actually calculate the likelihood that a virus will spread. And one of the key factors is how many people in the population are sensitive to the virus. And when you tip the scales so that there's a lot of immune people, it makes it harder and harder for the virus to spread. And so you can make a calculation given certain parameters, how many people have to be immune to the virus in order to stop it from spreading. And that is, you're right, that's herd immunity. Hopefully that threshold is, I encourage everybody to get the vaccine. We really need to get it above that threshold. If people have wild ideas that are unsubstantiated about the negative aspects of vaccines, certainly, yeah, the way that I think about it is, and so if you have friends or family members that are nervous about this vaccine, especially because it was, created very quickly, they've already tested each of these vaccines on at least 15,000 people. And so they can tell if among those people there are any bad effects. And so far, no bad effects. So that's reassuring. The other thing is that at the rates that this virus is spreading right now, you have, just an incredible number of people getting infected. And so we're kind of at the stage where either you get infected or you get the vaccine. We're kind of near that, right? And me personally, I would rather just have introduced into my body, a small mRNA molecule rather than a virus that has all these different proteins, has a ton of different, that mRNA molecule plus more mRNA molecules. And that just sounds way worse. And so I feel like if you're worried about side effects, the side effects of getting SARS-CoV-2 must be way worse than anything that this vaccine could do. And we know that claim is actually true, because 15,000 people have taken this and have not gotten bad side effects, whereas 15,000 people that have had SARS-CoV-2, they have this fatigue syndrome for a long time. Some feel this sort of neurological cloudiness for a long time. Of course, death is a very serious side effect, So I really encourage everybody to get the vaccine, and to just, if you guys know more about science and are more interested in this stuff than your parents probably, or your extended family. And so, just try to reason with them. My intuition is, or my instinct, not intuition, but instinct, is to just yell at my family and say, like, no, don't be stupid, like, do this, like, listen to me. But of course that goes nowhere. We know we can't just yell at people. So, reason with people. And if you are untrusting as well, just look at the data and see what comes out. Right now we only have press releases with some preliminary data. So it'll be important to actually look at the larger data, and that'll be made public. The vaccine companies have already said that they would do that. So I think I signed up for the Moderna trial to be a test subject. I didn't get chosen because I'm like sitting at home all the time and I'm not at risk of getting the disease. But I think it's pretty safe. Definitely. It seems like it should be.
Yeah, because this is embarrassing, but like I have TikTok, and like the number of people who are just like, don't get the vaccine or don't trust the results are like, oh, and I'm like, oh no, like,
Yeah.
like we need to get it. And I just like don't know how to react to like a good number of people being like, even if it is like substantiated, like I won't take it, because like that's what we're seeing right now. Like even though we should be social distancing and wearing masks, like there's clearly, like I went to Pacific Beach just to like get a takeout order, and it was as if there was no pandemic occurring, and people were just like shoulder to shoulder, no mask, and I'm like, oh God,
Yeah.
no, this is not what needs to be happening. I like haven't gone since because I've just been so scared.
Yeah. Well, I live over in North Park, and most of the time people are very responsible, but, and I just don't go out anymore, in the evenings or especially on the weekends, to the breweries or whatever. But every once in a while, we'll be walking the dog through that area, and it's like, you like see like these places packed, and you're like, we're like in a pandemic. What are people doing? So
Yes.
yeah,
Yeah.
what I've been telling people, now that we know that there's a vaccine, like we should just chill out for the next 6 months, until we get the vaccine. I know it sucks.
Yeah.
It definitely sucks. But
Hmm.
there's an end of this. And so let's
Hmm.
just limit the number of people that get sick until
Hmm.
that point. I mean, if it okay, if we thought it was gonna, so the last time a vaccine was made rapidly, it took 4 years. Now that was like in the 1960s or something
Hmm.
for measles. I'm gonna miss the details, but it was a long time ago. We were slow, and it took 4 years. Now this fact, so if it was going to take 4 years, I'd say, okay, well, I don't know. That's a really long time to just sit in my office here and give lectures.
Yeah.
And so I'd say, like, well, let's try to do some other mitigation strategies. But we have a vaccine,
Hmm.
so let's
Yeah.
just chill out. And then the other thing is, like, let's just treat this time as like a special moment in our life. It's a shitty moment, but it's special in that we're doing different things, right? We're not just
Hmm.
living our lives as we normally do. And so, like, in the end, we'll learn from this, right? We'll develop new hobbies. We'll develop new ways of interacting, or I don't know. I can already tell that in the future I'll be doing more Zooms with people that live on the East Coast and keep better in touch with them. So while I'm not keeping as good of in touch with people that are local, I am learning how to better, interact with people far away. So I think this is a time where we can just live life a little differently. It's like shitty vacation, you know, and then 6 months from now get back to normal. So.
Yeah, especially like over the summer, my friends and I were just joking, like, this is a self-care summer, like 4 months or like 6 months, like just self-care, just sleep in all day, take care of yourself. Whether that being like exercising or just eating ice cream, that depends on you. But like just like enjoying enjoying the year off. But yeah, I wanted to get your perspective on that. Thank you for answering.
Yeah, no worries. Cool. Yeah, so if you have any questions at all, feel free to email me, look up my lab. And we do tend to have a lot of undergrads that work in the lab. Right now, we're not accepting any new undergrads or any new researchers in the lab, just because the lab is only functioning at 25% of its capacity. But once things start opening up, if anybody's interested, let me know that you attended this thing in an email, and that'll sort of help me remember who you guys are. So yeah, I don't know. Anything else before I go? Cool. Okay. Well, thank you so much for this opportunity.
Thank you so much for speaking for us, Dr. Meyer. We appreciate your talk for us, and everything was very interesting to learn, and this discussion really helped us too. Thank you.
Thank you. Definitely.
Thank
You
you
guys
so
take care.
much.
Good luck with everything.
Thank you. Good night.
Bye. Good
Bye.
night.